Hello everyone. This section is for anabolism, the use of energy in biosynthesis. After learning this section, you should be able to discuss the principles that govern biosynthesis, know the nitrogen fixation, and should briefly describe the structure and mechanism of action of nitrogenous. The problem faced by all cells is how to make the many molecules they need as efficiently as possible. They have solved this problem by carrying out biosynthesis using a few basic principles. 6 are now briefly discussed. The first, large molecules are made from small molecules. The construction of large micromolecules from a few simple structure units saves much genetic storage capacity, biosynthetic raw material, and energy. A consideration of protein synthesis clarifies this. Proteins, whatever size, shape, or function, are made of only 20 common amino acids joined by peptide bonds. Second, many enzymes do double duty. Many enzymes are used for both catabolic and anabolic processes in pathways that are said to be anabolic, saving additional materials and energy. Third, some enzymes in anabolic pathways functions in one direction only. Although many steps of anabolic pathways are catalyzed by enzymes that act reversibly, some are not. These steps require the use of separate enzymes, one to catalyze the catabolic reaction, the other to catalyze the anabolic reaction. The use of two enzymes allows the intermittent regulation of catabolism and anabolism. Thus, catabolic and anabolic pathways are never identical, even though they share many enzymes. The fourth, anabolism consumes energy. Many reactions in anabolic pathways are endergonic and will not proceed in the direction of biosynthesis. Cells solve this problem by connecting endergonic biosynthetic reactions to the breakdown of ATP and other nucleoside triphosphate. When these two processes are coupled, the free energy made available during nucleoside triphosphate breakdown drives the biosynthetic reaction to completion. The fifth, catabolism and anabolism can be physically separated. Catabolic and anabolic pathways can be localized into distinct cellular compartments. It is a process called compartmentation. In eukaryotes, this is easily done because eukaryotic cells have numerous membrane-bound organelles that carry out specific functions. Compartmentation also occurs in bacterial and eukaryotic cells. For instance, carboxysomes separate carbon dioxide fixation from other processes in bacteria. Compartmentation makes it easier for catabolic and anabolic pathway to operate simultaneously yet independently. The last one, catabolism and anabolism often use different cofactors. Usually, catabolic oxidation produces NADH. It is a substrate for electron transport. In contrast, when an electron donor is needed during biosynthesis, NADPH often serves as the donor. In this section, we will focus on the biochemical of nitrogen fixation. It is a process that atmospheric molecular nitrogen is reduced to ammonia, which is then incorporated into amino acids. Let's see the nitrogen fixation. The reduction of atmospheric gaseous nitrogen to ammonia is called nitrogen fission. Only a few bacteria and archaea carry out nitrogen fixation. In the eukaryotic cells, completely lack this ability. Nitrogen fixation often occurs in the free-living chemotrophic bacteria and archaea. For example, Azotobacter, Calabacillar, Clostridium, and Methanococcus species, and also exists in bacteria living in symbiotic association with plants, such as lignums, for example, Rhizobium. And the cyanobacteria also have nitrogen fixation processes, such as Nostoc, Anabolism, and Trichodesminium species. These microbes play a critical role in the nitrogen cycle. They complete the cycle from nitrate to ammonia, where nitrogen, which has an intermediate oxidation state. Let's see the process of nitrogen fixation. The reduction of nitrogen to ammonia is catalyzed by the enzyme nitrogenase. It is thought that nitrogen is reduced by 2 electron additions in a way similar to that illustrated in the picture on right. The reduction of molecular nitrogen to ammonia is quite exergonic, but the reaction has a high activation energy because molecular nitrogen is an unreactive gas with a triple bond connecting the 2 nitrogen atoms. Therefore, nitrogen reduction is expensive, requiring a large ATP expenditure, at least 8 electrons and 16 ATP molecules. Ferredoxin is used as an electron donor. Nitrogenase is a complex enzyme consisting of 2 major protein components. You can look them on the picture. A molybdenum ion protein. 这个重来哈。Nitrogenase is a complex enzyme consisting of 2 major protein components. A molybdenum ion protein joined with 1 or 2 iron proteins. You can see them on the picture. The molybdenum ion protein has an organic cofactor that contains molybdenum ion and sulfur atoms, whereas the ion protein associated with ion atoms. Ion proteins is first reduced by ferredoxin and then by ATP. ATP binding changes the conformation of the ion protein and lowers its reduction potential, enabling it to reduce the molybdenum ion protein. ATP is hydrolyzed when this electron transfer occurs. Finally, reduced molybdenum ion proton donates electrons to atomic nitrogen. Nitrogenous is quite sensitive to oxygen and must be protected from oxygen inactivation within the cell. Microbes use a variety of strategies to protect nitrogenous. The reduction of nitrogen to ammonia occurs in 3 steps, each of which requires an electron pair. 6 electron transfers take place, and these use a total of 12 ATP per nitrogen reduced. The overall process actually requires at least 8 electrons and 16 ATP because nitrogenous also catalyzes an additional reaction, then short-circuits the nitrogen fixation process. The addition reaction is the reduction of protons to hydrogen. The hydrogen then reacts with diamine to regenerate nitrogen. Thus, nitrogen fixation becomes even more expensive. Symbiotic nitrogen fixation bacteria can consume about 20% of the ATP produced by the host plant. Once molecular nitrogen has been reduced to ammonia, the ammonia can be incorporated into organic compounds. Okay, that's all for this section. Here is a word list for you.